Thank you very much. This has been a fun week so far. It continues to be fun. I've never been to Berlin, wonderful city. This is a great research center, and the topic that is near and dear to my heart, which is basis of infection. And so this Spring Academy is stuff that I love to talk about. I'm going to give an empirical example of influenza. I'm going to talk about an ongoing project that just started. It's a 5-year project that's just a few months old on swine influenza. But I'm going to talk about other influenzas as well. And I'm going to talk about some of the kinds of methods that we're using in this study, as well as some more general methods in the fields of health and medical geography, as well as spatial epidemiology, which sort of do both. So I'm also going to incorporate genetics into my discussion of pandemic influenzas. I'm going to talk about mostly pathogen genetics. I'll give you some examples of what I mean by that. And a lot of times, evolution of organisms is driven by place, by landscape level factors. So I'm going to talk about something called landscape genetics. So this is not the entire team of people that work on influenza, but this is sort of a core group, including Varun, who is in this room, and Henry Wan, who I've worked with on influenza for a long time, mostly on avian influenza, but this is a joint project with him. Richard Webby, who's at St. Jude in Memphis, and he's the director of the US WHO Collaborating Center for Influenza and Animals and Birds. Tom Liberto, who is basically the head of sort of wildlife diseases for the US Department of Agriculture. And Jim Lowe, who knows more about pigs than anybody ever met. He's a vet at University of Illinois. But I'm going to take a step back, and I'm going to talk a little bit about sort of this medical and health and medical geography framework. And there are different sort of theoretical areas within this field. And one of them is called disease ecology, which I'm sort of part of. And disease ecology is much bigger than geography, but this is the geographic part of the field of disease ecology, and it's very holistic. And my late colleague Melinda Mead came up with something called the human ecology of disease, and it's a sort of a heuristic framework to sort of understand health and disease. And this is, there are 3 vertices in this, of the triangle, and population, habitat, and behavior. Habitat is a synonym of environment, and so the population vertex is made up of 3 different categories, and sex, age, and genetics, and that's really about people. And then habitat's really about environment, and a holistic environment, and natural and built and social environment. The last is behavior, and so that's about social behavior, societal behavior, and then technology. And let me go into a little bit further detail on that. So I'll give you some examples of this heuristic framework. In the population vertex, we've got sex, so something that would be related to a disease that maybe women are more prone to get. So, for instance, HIV in Southern Africa, women get it more than men. And there are reasons for that. There's social reasons for that. Okay, married women especially. Age, look at the coronavirus. I mean, it was no surprise that most of the people, the older you are, the more likely it is you are to die. That has been the case for all coronaviruses, not just this SARS-CoV-2. So people who knew coronaviruses knew the age distribution, what it was going to be. Okay? They knew young children were, almost none of them are going to die. So our immune systems, as we get older, I'm still pretty young, so my immune system's but you know, they don't do as well. So there are some genetic factors. There's a few listed there, but I'll think of one. So BRCA is a gene that it's a mutation that that is associated, highly associated with breast cancer. So that is a sort of a genetic factor at the individual level. Okay. So the habitat or environment protects. So there are natural sort of biophysical factors that can be associated with health and disease. Think about diseases that I study, malaria and cholera. They're both highly related to the natural environment, to rainfall and temperature, things like that, right? There's a lot of different aquatic environmental, factors associated with cholera, and malaria is related to rainfall and temperature and things like that. So you think about HIV, it's a socially driven disease. It's a sexually transmitted infection mainly, and so social factors, are related. And then you think about things that are related to overweight and obesity and the built environment, right? Berlin is great for me because I walk a lot and go to the public transport. In North Carolina, where I live, not so good, right? I have to try to exercise. Here, I'm just getting exercise. And it is dark late, which is nice too. So this, the last vertice is behavior. And so there are things like, marriage and norms and sex norms and things like that, are belief systems that will either protect you or put you at risk of different sort of diseases, right? Social organization, you think about coronavirus and, the fact that I move around a lot in this world and I'm not the only one. And so, the way we're organized nowadays in a global, world, and we move, we learned about that yesterday. We have, sort of technologies that we've developed, like vaccination for coronavirus. And some people, even though this is, probably the most extraordinary set of vaccines that have ever been developed, because of politics and sort of, other kinds of belief systems, then, the vaccinations those technologies aren't always taken up. Technologies such as sanitation are really important to stop the diarrheal diseases that I study, like cholera, right? Now we have a malaria vaccine as well. So anyways, so this is, you can think about this as like, okay, that's just all over the map. That is sort of, maybe too holistic, but it's a framework to allow us to start thinking about health and disease more holistically, interdisciplinary, transdisciplinary ways, okay? So in addition to sort of this theoretical context in this field of, health and medical geography and spatial epidemiology, we also we're in sort of this technological revolution. So we all have, every one of you has a GPS in your pocket with your phone, but we have all kinds of different devices, and it's really easy to collect spatial information, maybe a little bit more difficult to know what to do with it. But we're in this big data era of spatial information. And there's just a whole bunch of different kinds of GPSs shown there. Some of them get you down to the decimeter. Some of them track you for long periods of time, like that GPS tracker. And then we have, software and programming languages that allow us to, geographic information software and sort of R code that allow us to do spatial analysis and integrate data sets together. And then integrate them with things like satellite imagery. So, in the bottom right-hand corner, that's a Landsat satellite imagery. And in the bottom left is a Google Earth image, which is a satellite image. So we have all this sort of rich, big spatial data. But probably, well, I'll say definitely more importantly, we have many, many, many decades of empirical and theoretical work. Others have done work before us. Okay, these are just a few people: Michael Goodchild, Waldo Tobler, Tobler's first law of geography. That is, things that are further apart or more different. Anyways, that Peter Haggett, who's done a lot of work on spatial network analysis of infectious diseases over the years. And those are NAS member, that means National Academy of Science members. And so then there's a few folks at the bottom that I could have chosen others as well. Subbu Subramanian is a geographer at Harvard who does all kinds of sort of, econometric modeling and spatial analysis. And Sarah McLafferty is at Illinois, Dan Brown's at Washington, and Andy Cliff is Peter Haggett. So anyways, we've got this rich theory. So read, because we've got incredible amounts of work done before us. Okay, this slide is sort of hot off the press. Nobody's ever seen this before. It is a brand new study that literally, it actually isn't even officially funded yet. So this is a center that we're going to start at University of North Carolina, Duke University, and NC State University. And it's a very low bar for what we're going to try to do. And so this is a National Science Foundation grant this is the grand challenge listed there is what we're going to be doing for the next few years. And that is, we want to be able to characterize the pandemic potential of an early stage disease outbreak and ready population health models for mitigation before that pathogen spreads regionally or internationally. That's easy, right? So how are we going to do that? We're going to do that by we have this sort of leadership team at the different universities, and we're going to form working groups. And we have sort of 3 questions with these working groups. And the point of this slide is that we are going to try to do pandemic preparedness work, and it's extraordinarily interdisciplinary, right? So the first is, how do behavioral feedbacks and geography impact the spread of infectious disease on contact networks? And you can see that the disciplines that are included at those 3 universities, including mine, are economics, geography, computer science, network science, epidemiology, evolutionary anthropology, social medicine, bioinformatics, and statistics. Okay. The second is, how does a network science approach inform understanding of outbreak outcomes? So again, lots of different disciplines: network science, biodynamics, statistics, geography, global health, biomedical engineering, communications, and disease ecology, which is interdisciplinary in itself. Okay, and the last question is, I can't quite read. I got to walk over here, is what new data are needed to understand early stage outbreaks. So how can we, and we're going to leverage our partnerships and our existing studies to figure out what we need to do, okay? Including the one I'm going to talk about today. So this is not completely, this is very much related. And we're going to pilot test data we already have or are collecting, like this swine flu study that I'm going to talk about today. And then we are going to form a center called the Triangle Center for Predictive Pandemic Intelligence. TRICEPI. I don't know, that's kind of a weird, we can help us with our acronym. So anyways, this is sort of maybe pie in the sky, but we will see, because this is a quagmire to be able to do this, to do that grand challenge, right? But there's no question, and you all are from lots of different disciplines, and and this is going to be tough work. The first part of this kind of work is to try to understand each other's languages, right? And so anyways, we'll let you know. Okay, so I'm going to start talking about viruses now, okay? Yesterday, we learned about One Health. We've unfortunately learned about bat-borne viral viruses these last 2 years, okay? We learned about coronavirus, and we already knew about coronaviruses, we just didn't pay too much attention, okay? So there's a pandemic of MERS, which is Middle Eastern Respiratory Syndrome, and there's an African tomb bat reservoir, an intermediate host, which is a camel, and it's transmitted to humans. Somebody mentioned this MERS the last couple of days. We have SARS, which was in 2003, horseshoe bats. There was an intermediate host, maybe one of many, which was, palm civets, and is transmitted to humans. Animals are very important to the ecologies of a lot of viruses, okay? SADS is just a swine virus, and it's a diarrheal disease actually in swine, and that horseshoe bats are also the reservoir. SARS-CoV-2, horseshoe bats, an unknown intermediate host. We didn't know what the intermediate host or hosts for SARS were for a while, and so there probably are some. I mean, there was massive killing of palm civets in China for when there was SARS, and that helped. In retrospect, it helped. These are in the sort of wet markets, as they call them, right? Which are just live animal, fresh food. Nipah virus is another fruit bat. Is a fruit bat, and it goes into pigs and then humans. And then Ebola, we learned about that. That's also got it's a bat virus, and there's an unknown intermediate host, and it's transmitted to humans. So these ecologies are complex, and it takes a while to figure them out. Here's some scary pictures of these things. The horseshoe bats that is SARS-CoV-1, the reservoir. That's where it came from. And by the way, there are thousands of coronaviruses in bats, right? There are thousands of coronaviruses in bats, and so it's not like we can actually find them all, and so that's why we're trying to come up with a system to intervene after the outbreak, because there's no way we'll be able to figure them out all before. So each bat has many coronaviruses, and then novel coronaviruses. Okay, so on the right is the civet, which was the intermediate host, and so it was found to be associated with SARS-CoV-1. Okay, so I am going to talk a little bit about some of the things that were talked about yesterday here. And this, you all know some of this terminology, right? So this is the, the phylogenetic tree on the left, which is, and Adrian talked about GISA data yesterday. And so that's just from the other day, a snapshot of the history of SARS-CoV-2 virus and this family tree, right, on the left. And then on the right is a snapshot of a map with sort of pie charts of the dominant clade, right? So that's some terminology. Clade is sort of Omicron, is a clade, right? And there's different subvariants of Omicron. Well, there's different nomenclature. Some of them are numeric, and those are sort of those names are and so the one thing I'm going to talk about for the next 40 minutes is incorporating biomarker data, so into social data and other kinds of data, right? Incorporating them sort of a biosocial approach. And so there's a reason why distributions are what they are, okay? And that's something that we can try to figure out why, okay? There's a field that is called landscape genetics. It's an interdisciplinary field that's premised on the idea that exploring the spatial variation in genetics can illuminate how organisms exist in and move across the landscape, any organism, any living organism. There's two steps to landscape genetics analysis. And the first is to identify patterns, so how genetic characteristics vary in space. And then the second is process to correlate those patterns with specific characteristics of the underlying landscape. And so the goal is to identify landscape factors that could be, anything in that triangle of human ecology that I showed you, and that might be driving evolutionary processes of an organism. Okay? Human, but I focus on pathogens. So we can use landscape genetics, to study infectious diseases, and we can use landscape genetics to do One Health studies that look at that ask questions about the human, animal, natural landscapes, right? And how that might drive disease emergence and pathogen evolution. We can ask questions like, what local level population environmental drivers are related to molecular evolution of pathogens, the change in pathogens, or act as barriers to the spread of those pathogens, or drive diffusion of new variants, or what's the impact on vaccination campaigns on those. So I said we live in a spatial big data era. We've gone further. That's John Snow's map that we heard about yesterday as well. We've come a long way with all these spatial technologies, but we've also come a long way with sort of genetic and biomarker technology, right? So, and these like things like PCR, which, to characterize the genetics. And it's enabled the integration and analysis of spatially referenced geographic descriptions of pathogens. And so we're in a big spatial data and a big genetic data era. So that map on the right is actually a map of Falciparum malaria in the Congo, in the Democratic Republic of Congo. And those, that's a surface map, in the red areas, that's like 85% of the people in those areas have malaria at any given time, right? And then, goes down from there. But so there's some really hot spots of malaria. And so how do we do that? Well, we randomly selected blood spots from tens of thousands of people around the country through demographic and health surveys. It's mainly so blood spot, filter paper, it's for HIV testing. We take one of the blood spots and punch it out on the right? And we take it back to North Carolina, and then we can look at things related to that. But it's spatially referenced, right? We've got tens of thousands of these, and they're random. And so we actually have a couple of different cross sections. One's 2007, one's 2013 and 14, and we can do things like this. So we can not just look at the distribution of malaria, but we can actually look at mutations associated with drug resistance. So there's a drug called SP, it's widely used in the Congo, and in those red areas, there's a drug resistance mutation for that particular drug. And so in those areas, the parasites are resistant to SP, and so we have to use other drugs, including ACT is another drug. So there is another thing that we've done in the Congo. You guys will recognize that kind of test. I think most everybody took one this morning, and it is a rapid test, right? And it looks exactly the same as a coronavirus rapid test or a pregnancy test. And so 2/3 or more of malaria diagnoses in Africa are done with these rapid tests, right? And so it's targeting a particular gene, and it's called HRB2, and sometimes there's a deletion to that, in the pathogen, and you get a false negative. So if you get a false negative, then you don't get drugs and you're not treated. And this could be a huge problem. Okay, and a serious threat to malaria elimination, right? And so we took, this is kind of an old slide, but we have a much larger study now. But so we found, though, this is a random selection of those blood spots, and we found that 6% of all of the infections in the Congo, 6% have HRB2 deletions, and so we'll get false negatives. This is a huge problem, but we're trying to use a spatial approach to understand sort of the ecological drivers of that diagnostic resistance. Okay, and there actually is that's just kind of we have an individual level, sort of village level data, but this is just an overview map, and so North and South Kivu and over to Rwanda and Burundi is very high, and then Kinshasa is very high, and then there's some other areas where it's very low. It's more common in low malaria transmission areas. So we can actually understand the drivers of that diagnostics, but it can also be used for practical reasons, right? We can actually know where we might actually have to go old school and use microscopy to do diagnosis, right? Now I'm going to move to flu, and I am going to first talk about some of the sort of ecology of flu. So influenza viruses, they cross species barriers through close contact with infected birds, in particular domestic poultry. So you can see in the green birds are domestic poultry, and you'll recognize some of the H1N1 things like that. And when people have contact, or other wild birds or other mammals have contact with domestic poultry, there can be interspecies transmission of influenza. So Robert Webster at St. Jude also was the one. He went around the world. I think he's in his 90s now, and he went around the world and collected flu samples from all kinds of organisms, including whales. Anyways, and so there's some transmission of influenza from wild migratory birds to domestic or captive mammals as well. So it's a complicated ecology, and pigs are a perfect mixing vessel for these different viruses, and it has to do with the epithelial cells of their respiratory systems. They're similar to humans, and so there's there's mixture of genetic material and genetic shifts, and so we can have combinations of viruses that are bird viruses, both wild and domestic bird, and pig viruses and human viruses, and it all mixes, right? And pigs are a wonderful mixing vessel. So we've heard about all of these flu pandemics. These are the last 4 flu pandemics. The Spanish flu, the so-called Spanish flu in 1918 was an H1N1, and then the so-called Asian flu and Hong Kong flus, which are H2N2 and H3N2, and then 2009 swine flu. We got away from place names because of the stigma attached to them, and now we call them other things. So anyways, and it's an H1N1 that is related to the 1918 Spanish flu as well. The 1918 H1N1 pandemic contributed virus segments to the last 3 pandemics. Okay, so this is all changing over time. It's all interrelated. So this is from a paper that Richard Webby, who works on this project, created, and I'm going to give you a little background about avian influenza. So the country shaded in red, and H5N1 avian influenza in particular. So this is very relevant to pandemic preparedness, because this is the one that everybody was really worried about, okay? Because it's highly virulent. It kills a lot of people. It's not very transmissible. So the question was, if it's not very contagious. The question was, oh my God, if a H5N1 becomes more transmissible and becomes a respiratory infection, we're in trouble. So that is the nightmare scenario. So anyways, those red countries are, where there's H5N1 outbreaks in domestic poultry and humans. The blue are just in wild avian species. And by the way, it's been spreading this year a lot, and last year around the world. And there's, additional, the purple ones are like the mammalian species that's been found in Indonesia and Vietnam, pigs, and then I think in Egypt is donkeys. And so anyways, there's that's the sort of distribution of H5N1. So what we did is we looked at sort of this, you'll recognize that sort of, human ecology or disease approach. And we studied what elements of the social and natural environments, and this is in Vietnam, were driving the H5N1 evolution, like change in genetics. It's an anthropozoonotic disease, which means humans and animals. And the majority of infected birds in Vietnam are domesticated birds and not wild birds. And they're highly mediated by humans. Humans are part of the ecosystem that's driving this, right? We're the ones, eating all these chickens and ducks. And so the goal of this study was to understand the molecular change of H5N1 avian influenza viruses as the outcome of interacting environmental and social pressures on the hypothesized molecular change. So in that triangle, again, you can see a lot of human variables, like human socioeconomic status. In Indonesia, for instance, when there was, H5N1 was floating around, poor people got it more than rich people. And why? Because their chickens were sick and they were poor, so they kept them. They didn't kill them, right? Things like that. So humans are part of this ecosystem. So from that study, there's a lot of findings that we have. There's a bunch of papers listed at the bottom, but some of the highlights are that evolution of H5N1, in domestic poultry is highly correlated with the location and the spread in geographic areas. So that second bullet point is the correlation varies by time with a classic isolation by distance pattern. Isolation by distance is Darwinian theory, and it just means that, like the finches in the Galapagos Islands, the morphology of their beaks was different the further away, but actually the local landscape actually changed the beaks as well. So isolation by distance is you get further away, there's genetically more dissimilar. So we found that in Vietnam. Variables that are related to both the environment and social ecology of birds and humans interact to affect the characteristics. So we can actually predict the sort of genetic character of viruses. So a combination of suitable virus for species mixing, humans and birds, and high numbers of densities of birds and people, those were sort of the drivers of the H5N1. I'm going to switch to swine flu, which was what I started with telling you I was going to talk about, and which is this ongoing study, okay, or just begun a few months ago, and it's a U.S. and China study. It's in both, which is the largest swine industries in the world, so our collaborators there wrote this paper back in 2021. And they found this recently emerged genotype reassortment of an avian-like H1N1 virus. They also looked at swine workers, and they found that 10% of them were positive for the virus. So humans are actually getting it from swine, and it's going both directions, actually. This greatly increases the opportunity for virus adaptation in humans, and so raises concern for pandemic viruses. That's just a phylogenetic tree of this H1N1 from 2011 to 2018, and that the bottom part is just the diversity of the virus and the change. So in this present study, we have a few hypotheses, and one is that novel influenza viruses continue to emerge and evolve to be highly transmissible viruses. That there's also intercontinental movement of genetic segments in the US and China, and that's going to further diversify the swine influenza viruses and facilitate the emergence of novel swine viruses, right? And so we think that we can actually characterize the ecology of the swine industries to sort of understand and predict, the evolutionary dynamics of swine influenza virus. Okay, so China, 46% of global production, the US, 11% of the global production. Basically, almost all pigs in the US are made through factory farms, right? There is, they're factories. There's like a Ford motor plant, right? And in China, it's heading in that direction too. Very large percentage. This is sort of old data, and it's just much higher. So anyways, farms are getting big. It's a commercial operation. In China, they don't have wild pigs. In the US, we have feral swine populations, more than 6 million of them running around, and sometimes they get into the swine facilities, right? And in North Carolina, where we live, we have them. In Illinois, which is another one of our study areas, we don't. So this is just another graphic of showing the swine industry as it grows, and this is in the US, and the size of the farm getting bigger and bigger. There's basically no such thing as a small family farm that is meaningful at all. And the same thing is going to be true in China as well. So the swine, it's a factory. And so they only live 6 months. And about 6% of them die during that 6 months, right? And the idea is to make them fat and get, bacon, right? But they're closed systems, right? They're closed systems. And so that the figure on the top basically shows, going from farm to farm. Sometimes they move 3 times, owned by the same company. They move from farm to farm. The bottom graphic is actually data from Richard Webby and Jim Lowe, work that they did in 4 different places in Illinois, Georgia, Oklahoma, and Nebraska, A, B, C, and D there. And then the different colors and sizes are actually the different influenza A subtypes, like H1N1, H1N2. And looking at those colors, and you don't have to do any spatial or cluster analysis. It was quantitatively, you can see that there is clustering of different pathogens subtypes within different levels of the production, as well as within different areas. Okay. In this study, this is a picture of a Varun in January in Illinois. He was chasing down pigs. One of them kicked him in the side and taking nasal swabs, which we've all had now because of coronavirus. And we're collecting thousands and thousands of nasal swabs and then isolating the viruses. And we're going to find thousands and thousands of viruses and also reassortment events. Okay, the reassortments is what we're focused on. Find those reassortment events, and then we're going to try to figure out which ones are sustained and which ones stick, and what ecological circumstances are related to the ones that stick. Okay, they could potentially become, the pandemic swine viruses, right? It's not just about humans, it's about production as well, and huge economic losses in these industries. And so those are the 2 kind of locations in the US, in North Carolina and Illinois, that we're collecting data, and then in China, which hasn't started, and those are the locations in China that our collaborators are going to collect data. This is just from the Midwest, and this is just some data that we collected. This is sort of preliminary data that we haven't collected prospectively, but Jim Lowe and his group has collected over the last few years, and there's some diversity of subtype. That's what this graphic is showing. You can see that the orange one is H1N1, H1N2. Okay, so there's some diversity of subtype, and there's also some differences in magnitude as well. So this is a map of the swine systems. The different colors, A, B, C, D, and E, are the different companies. There's names of the companies, right? And they're different swine farming systems that are supposed to be closed systems. Okay, the pigs go from farm to farm, but within the same company. And so they're interconnected. And then the swine move across during their 6-month life cycle. And so we are collecting, like Varun was doing, virus sequence data, and then we're going to measure the impact of ecology on gene flow and, again, reassortments. So all these clades and all these names, they mean something, okay? So the frequency on the right is for all of that data that, those locations I just showed you. These are we claded them, okay? And those are the frequencies. So gamma is the 36% frequency. Delta 1 is 28% of the frequency. So gamma is a classical swine flu lineage, the result of a triple reassortment event between H3N2, H1N2, and H1N1 viruses, right? Delta 1 is seasonal human flu lineage, emerged from humans, an introduction to swine in the early 2000. It contains both H1N1 and H1N2. Delta 2 is a seasonal human flu lineage that contains H1 and N2 genes, and it's very different. It's diverged away from Delta 1. NPDM is the 2009 pandemic swine flu that actually has some relevance for coronavirus, because in Europe they spent about €1 billion to develop to buy a vaccine, and nobody really used it because it didn't kill very many people. And so maybe that's one of the reasons why people have a little bit of their guard down for this coronavirus, is because they got burned with spending €1 billion. But anyways, that's 10% of the swine flu lineage, okay? And then alpha is a classical lineage related to 1918 flu. Beta is a classical lineage. It's a small percentage, but due to sort of reassortments between a few different strains. Okay. This is a graphic of the distribution of the percentage over time of these different clades, okay? So the main takeaway here is, while some clades were persistent across that 15-year period that's shown in there, some of them emerged and re-emerged later. There are, the past, 2 years, the most common clade is Delta 1 and Delta 2 and gamma, with a slight detection of alpha and NPDM. So those are, the proportions change, and some of them go away and some of them go away forever, and some of them, which ones are persistent is what we're going to try to get at. And can we predict it? So that's just a phylogenetic tree. One phylogenetic tree of many. These are probabilistic things, and so that's actually the same data that we're just visualizing it in different ways. So this is this sort of family tree, one possible family tree, and okay, and so I've mapped it here. I meaning Varun mapped it here. He did this in Thailand the other day, and so we have these different clades. The one at the top, these two are the most dominant clades, and so this just shows the spatial distribution, and the source is the system, the different companies, and then and so based on this distribution, we observed that there's strong clustering by the different company, right? The different source, right? So they're clustered within the different swine companies, right? Production companies. Okay, so there's a question of whether or not, and you know, whether or not there is an isolation by distance, whether if there's correlation between the different swine systems, right? Then maybe there's some gene flow between systems. Maybe they're really not closed systems after all, right? And so so, you know, this is both in time and space. Are they different in time and space, right? So this is the next two the next two most common ones, delta 2 and NPDM, and NPDM is a concern because that is the 2009 human, you know, pandemic, the one that presumably started in Mexico and went to the US and then spread around the world. So anyways, this is just, you know, showing the, you know, the different, you know, systems. This is, these are sort of the more minor ones as well, and you can't really see much from those patterns as well. Anyways, so what I keep on talking about sort of this, you know, ecological perspective and what we're going to do in Vietnam with H5N1. We got viruses from all over the country, and we looked at human and environment, you know, this is sort of backyard poultry, and we had some human, you know, cases as well, not very many, and so there are all these different sort of multi-level factors that might be part of the ecology that might be driving it. So which ones are important? Okay, so pig level variables like sex and the amount of pigs are really snotty, as Varun well knows. Anyways, and so, you know, some of them are sicker than others, right? And so, you know, what source farm do they come from? The number and pen level. So these farms are sort of spatially distributed in, you know, pens. And so that some pigs hang out together and then other pigs hang out together. And so there are pen level variables as well. Some of them are bigger, some of them are smaller. In Illinois, they get heated, there's heaters and things like this, right? So then there's barn, site, farm level variables, and they're moving. So things like, you know, if they move a long way, right? Maybe they're going to be stressed out. Actually, literally, that's what the pig people tell us, like Jim Lowe, is they actually get stressed out and physically, you know, so that might be making them sicker, right? And so there's lots of, this is like a laundry list of variables. And so it's about pig, you know, barn site level variables, as well as, you know, pig flow between those sites. And then we have sort of more regional level and neighborhood level and county level and state level variables, things that are, you know, like the temperature in those areas. Illinois is really cold in the winter, pretty warm in the summer. North Carolina, not that cold in the winter. And then North Carolina has proximity to feral hogs as well. We have a lot of measurement going on. These places, these are again factories, they're high temp. There's like, you know, temperature and humidity and ventilation controls, but there's data collection associated with it. And the farmers don't use this data except for to monitor their pigs, but they collect it and we take it, right? And we also have pig level variables like whether they're vaccinated and things like that, right? So this Tom is again the keeper of all of the feral hog data in the country. And so in the upper left-hand corner, that map shows the feral swine distribution sort of the pig production overlaid with the sort of the domestic pig production. So the green is the feral hog distribution, and the orange is the sort of pig production distribution. And so feral swine in the US have been exposed to both avian and swine influenza viruses, and they have these swine influenza. And so the three other maps are basically just 2011, 2012, and 2013 of whether or not these feral hogs, the way they collected these data is they shoot them, they kill them, and then they collect the virus. They're pests, right? I mean, there's some of them, sometimes they do it by helicopter. Anyways, they just destroy farms. But anyways, so anyways, there are, this is something that we're going to be sequencing them and to see whether or not there's gene flow from feral hogs, and we don't really know the answer to that question. Just about done here, but how are we going to do this? Well, we're going to be using sort of deep learning, machine learning methods to measure the probability that a novel sort of reassortment emerges and becomes enzootic. Okay. And then this emergent risk, we're going to be looking at the sort of genetic pool and how it changes, and that graphic that I showed you of like different clades, we can actually dig down much deeper than clade. We got the subtypes, H1N1, we've got the clades, it goes down deeper, but we're looking at full genomic sequences of these viruses. So we can slice and dice it in very detailed ways, and then we can look at and correlate that with the human environment ecological setting, right? And so these deep learning methods are going to be used to sort of extrapolate virus emergence and the trajectory dynamics of these novel swine influenza viruses. Okay, my collaborator Henry Wan is an expert in he's got teams of people doing this stuff. I'm not a machine learning person myself. So I'm going to come back to this slide, and I'm going to say that what I just brought you through was sort of a very ecological approach to looking at flu viruses. But there's a lot more, and you all are from different disciplines and different methodological backgrounds. And I'm going to say what I said at the beginning, which is there needs to be more than just that team of people with the expertise that we have in our group on this swine influenza virus to be able to do what we say we're going to try to do in that grand challenge, and which is to characterize pandemic potential in an early stage disease outbreak and ready population health models for mitigation before that pathogen spreads regionally or internationally. We know it's going to happen again. There's no question about it. I was asked to send you some reading material, and I gave a couple of chapters of my book. At the end of the chapter on emerging infectious diseases, I'm not prophetic. I said there's going to be a coronavirus pandemic, and I wrote that in 2007. That's not me. We knew that there was going to be another coronavirus pandemic, right? So how are we going to stop the next one? And so we've got behavioral economists, we've got an entire group of network analysts, sort of mathematician types, but then we have sort of bioinformatics folks that do this sort of genetic stuff. Anyways, I don't know the answer to this question. We got like $1.5 million to play with for the next year and a half, and then if we do well, they're going to give us 30 million. That's a drop in the bucket to be able to do this work, but we're going to do our best. I'm going to end there, and I'm going to, this is my research group that Oliver mentioned. the Spatial Health Research Group at UNC, and we're also part of a larger group at UNC called IDEAL, which is the Infectious Disease Ecology and Evolution Lab, which is a whole bunch of people that do infectious disease work and lots of different infectious diseases. But there's our website. The funding for that swine flu study, the funding for the other studies is down below. But I will open it for any and all questions. Thank you very much.